Hi, my name's Malik Appleton. I'm a fourth-year student here at the University of Kentucky, actually first year in the PharmD program at the College of Pharmacy. And I'm working in Dr. Keith Pennypacker's lab in the Center of Advanced Translational Stroke Science in the College of Medicine. And the title of my project is Cellular Differences in Male and Female Rat Stroke Immune Responses. So stroke is actually the second leading cause of death in the entire world, right behind heart disease. And ischemic stroke, the type that we focus on in my lab, actually occurs when there's a blockage in a main artery of the brain. When this blockage actually occurs, there ends up being cell death in the brain due to the blockage of their blood supply, so they don't get their nutrients or oxygen. And when the cells start to die, they actually start to release a sort of alarm for the body to begin its immune response, which is typically hallmarked by inflammation. And inflammation is actually not good for the brain at all. It actually causes further damage after stroke, and that's one of the main reasons why people are having problems in their long-term recoveries. They're not able to get their communication skills back. They're not able to get their movement back because of that further damage in the brain after the stroke because of inflammation. So my lab, we actually want to focus on reducing that to improve longer-term outcomes. So we actually found this molecule called leukemia inhibitory factor, or LIF. It's a protein that in few studies has been shown to improve longer-term outcomes after stroke in young rats by actually reduces the amount of swelling in their brains, reduces the infarct or the hole they have in their brains after the stroke occurs, and the rats are actually interacting with the other rats and moving better longer-term after that. But of course, with all good science, you need to check all your variables. So what we wanted to do was do a study, looking at other things. We wanted to look at how older rats were affected, and we wanted to look at the gender differences between male and female. So that's exactly what we did. So with these, we got older rats. We had male and female, and what we did was we sedated them, and then we subject them to a surgery called MCAO, middle cerebral arterial occlusion, where we would go in and we would block the middle cerebral artery of these rats in order to induce a stroke. And then after that, we would do injections at 6, 24, and 48 hours after the stroke with either PBS, which was our buffer solution as a control, or we would inject them with the LAF, which was the target molecule, of course. And then after the 48-hour time point, we did MRI scans on the rats' brains for other testing, and then we collected the brain, of course, because that's where the stroke occurred. We collected the blood, because that's where the immune cells traveled to the brain in order to begin the inflammation. And then we also collected the spleen of each of these rats, because the spleen is well known to actually facilitate a lot of the immune response after a stroke occurs. The cells will migrate from the spleen and go to the brain through the blood. So here we actually have some of our results. This is just a snapshot. We looked at we used flow cytometry in order to look at the number and types of cells in each of the samples. And we actually looked at B cells, CD4 and CD8 T cells, and macrophages and monocytes. And we had the 4 treatment groups of male PBS, female PBS, male LIF, and female LIF. And you can actually see some sort of visual trends on the graph that show some interesting data. However, you can't really make any conclusions on this because we only had 4 rats per treatment group, so there was some outlier effect, and that's why we have very large error bars with a lot of our results right here. So it was kind of more of a pilot test just for me it was me onboarding with the lab. This was my learning experience for the summer. So I was more getting acquainted with flow cytometry and the actual protocol of doing all this, and working with the rats themselves. That was a whole nother thing. And so the main idea of this is that we want to do some future testing with this and actually test more rats and test for more immune cells. And we actually think that we could get some better data in the future, and eventually this could even become a clinical trial for human stroke treatments, because rats have very similar anatomies and physiologies to ours. They're mammals, and it's easy to map these types of conditions in them, and then they can be translated directly back to us. So hopefully this could even be a potential stroke treatment in the future. Thank you for your time.